So in this experiment, we're just going to be looking very simply at general microscope use. And to begin with, what you're going to see is I'm going to put the microscope around to the smallest pair of objective lens, which is the 4 times magnification, and the eyepiece gives 10 times magnification to give a total of 40 times magnification. You can see the slide is clipped in place and the lamp is on to provide light up through the sample so that we can see it. I'll move the coarse focus wheel for large adjustments. I'll move the fine focus wheel for smaller adjustments. Once I'm pretty happy that I've gotten a good clear focus on the image at hand, I will then move slightly more powerful objective lens, either to 10 times or 40 times magnification, and then I'll do the exact same thing, move the focus wheel to the focus, and then I am done. Once we're finished with the general use microscope part, we're going to look at the plant cell onion investigation. So what we're going to do here is we're going to take an onion and we're going to get a knife, cut it up. Once it has been cut up, I'm going to then start trying to get a really thin layer of dermal tissue. It has to be very thin because light has to be able to pass through it, so that when we look at the sample under the microscope, we can see all of the cell structures such as the nucleus and the cell wall. What I'm going to do here is I'm going to get a forceps and try and peel a very thin layer because my fingers are too large. Once I have a thin layer, I can place it into the water. This to prevent that from drying out and curling up the top of itself. Then I go back and I try and get a few more slices. Once I'm pretty happy that I have enough and I've put them into the water, I'll then go back, I'll get my forceps and I'm going to take out one of those thin layers and I'm going to try and place it onto a glass slide. So you'll see here now, I have it on the glass slide now, and again, you can see it's very thin. I'm happy enough. Now it's time to get the cover slip. And once I have a cover slip, I can add it at a 45-degree angle to ensure there are no bubbles. But to ensure that the onion layer doesn't dry out, I'm just going to drop 1 or 2 drops of water onto it, and then I take my forceps and I'm going to add this cover slip at an angle of 45 degrees. 45 degrees because we don't want to trap any air bubbles, and the reason we add the cover slip is to protect the objective lens. We do not want to damage the objective lens at any point during this. So you can see I'm lowering it very slowly. Once it's on, this specimen is ready to go. So I place it onto the stage within the microscope. What I'm going to do here is make sure that the microscope is at the 4 times objective lens. I'm going to get down, have a little look through the eyepiece, and I'm going to start moving the focus wheel around until that cell comes into focus. And you can see it there now. Can't really see much cell organelles, but you can see the overall structure. So now what I have is I have iodine, and iodine is going to be used to stain these cells. So I'm going to drop 1 or 2 drops of iodine on the far side of the cover slip, and then what I'm going to do is I'm going to grab some filter paper and dab at the opposite side to where I just dropped the iodine. And the filter paper is great because it draws the iodine across the cover slip, and you'll see it here now in a second. It starts to draw the iodine across the glass slide through the cover slip, and as it does this, it's going to stain more of the cells. Once I'm pretty happy that there's no excess iodine there and that it's definitely come through the entire cover slip, I'm going to put it back on the stage, and again the same idea, I'm going to start moving around the focus wheels until I can get a clear image of the cells beneath. And you're going to see it now in a second. Once the stain has been added, you can clearly see nucleuses and the cytoplasm is stained as well. The final step of these microscope experiments then is to look at an animal cell, specifically a cheek cell. And the way we do this is we are going to take some cotton buds, and you then get the cotton bud and you swab against the inside cheek of your mouth. Now you do this a couple of times back and forth because you're trying to dislodge some cells. Apologies, it's not a great image, but it is what it is. It's what you have to do. Trying to get a few cells to break off from the inside of the cheek membrane onto the cotton bud, and then we just try to spread those now displaced cells onto the glass slide. Once we're pretty happy we've pushed any possible cells from the cotton bud onto the glass slide, we're going to go and take cover slip, and what we're going to do with the cover slip is we're going to add it at an angle of 45 degrees using our forceps. Again, cover slips 45 degrees to prevent air bubbles, and the cover slip is there to protect the objective lens from being damaged. Once the slide is ready to go, it's placed on the stage, and at 4 times magnification, the focus wheel is going to be moved until the cell comes into focus, and then if it's in focus, we move through the other powers of the objective lenses. Once we have done that though, there's not much you'll see without a stain, so the glass slide is going to come off, and what we're going to do is we're going to get methylene blue, and we're going to add some drops of methylene blue to this slide. This will stain human cells, which is really important here because you can't really see the human cells because they're so small and there's not much structure to them unless there's a stain added. So I get my filter paper, I draw the methylene blue across the cover slip just like I did with the plant cell. And once I'm pretty happy that the methylene blue has been drawn through and there's no excess, I place it back on this stage again, move through the objective lenses, make sure I'm using 4 times magnification, move the focus wheels about until I can get a clear image of the cell. Once I'm happy I've done that, I'm going to move to the slightly more powerful objective lens. In this case, it's going to be 10 times magnification. And at 10 times magnification, you'll get a much clearer image of cheek cells, which you'll see here at the end. So you can see the clear nucleus there, the cell membrane on the outside, and a stained cytoplasm as well.